Okay, it's Sneha Gangopadhyay, right? So,
Yes, sir.
okay, Sneha, so just introduce yourself in brief so we can talk further regarding your work experience, etc.
My name is Sneha Gangopadhyay. I am from West Bengal. I did my graduation in microbiology. I went on to do my post graduation in biotechnology. My hobbies include mandala painting.
Okay, Sneha, you did your MSc in biotechnology and UG in microbiology, right?
Yes, sir.
So currently, what are you
Yes,
doing,
sir.
Sneha? Doing some that is PhD or preparation for?
Yes, sir. Along with my preparation, I'm already prepared for and along with that, I'm also a creative writing mentor and a platform.
Okay, okay, okay. So Sneha, as you know, that is there are 10 unit syllabus of CBTM means biotechnology. So if I asked you among those 10 unit syllabus, what are the possible unit that is your favorite topics means in the CPTM syllabus?
Recombinant DNA technology.
RDT technology, only RDT technology, or some other subject like other microbiology, immunology, biotechnology, etc.
Immunology, I'm very bad at actually, sir. Uh, Maybe genetics and recombinant DNA technology.
Very good. So you are also interested in the genetics. Okay, Sneha, in genetics there are
Yes,
terms
sir.
like as a homozygous, heterozygous, hemizygous. What is the meaning of a hemizygous and homozygous?
Homozygous is wherein two alleles are same, like the altered forms of two alleles in a gene are same. Suppose let's consider a dwarf plant, as in T, which was considered by Mendel. It will be small t, small t. And in heterozygous, it will be like one of the character will be dominant, which is referred as capital T, and the other will be small t.
And what about the hemizygous?
So in heterozygous, we get, I'm not able to recall that. Yes, I read
Okay,
it, I forgot
okay,
actually.
okay, good, good. No issue. Moving ahead. Sneha, there are certain tests like the test cross, back cross, etc. So, what do you mean by the test cross and back cross?
In test course, what we do is we'll take any of the sample, which whose suppose we find, let's take an example. Suppose we find a plant, let's consider the example of Mendel's plant. If we want to see the character, suppose the plant is tall. Now we want to find out if it is heterozygous tall or is it like dominant tall, as in like we have homozygous tall as well, like which has both of them as capital T. So what we'll do is we'll do a test cross. So with the recessive uh, homozygous plant, we will do a cross. That is known as test cross.
Okay, Sneha, law of segregation is also known as the parity of gametes. Why it is known as parity of gametes?
Yes, sir. Since law of segregation tells us that each gamete will receive one allele from one parent. So that tells us that every allele is pure. Like whatever one receives from one offspring receives from their parent is pure.
Okay, there are two very similar terms that is known as the incomplete dominance and codominance. How you can differentiate between these two terms, that is codominance and incomplete dominance, with suitable examples?
Yes, sir. In case of incomplete dominance, what happens is the the parental phenotypes get mixed up and they get blended up and they give rise to a new phenotype. As in Mirabilis jalapa, which is one of the plant. One of the parents is red in color, the other parent is white in color. But they will develop a new phenotype in which the offspring will be pink in color. Like both the colors will get blended up and it will give rise to a new color. Now let's come to codominance. In case of codominance, what happens? The phenotype of both the parents will be expressed, as we can see in the ABO blood group system. If there is A and B, both kind of antigen are present on the RBCs, so both of them will be expressed in AB individual.
Okay, well done. Sneha, in ABO blood group system, there are three different types of a gene involved. That is I power A and I power B and I power O. So, what is the relation of that is A and B genes in between, and what is the relations of A and B to the reference of O?
IA and ID are codominant. So if they are both present in an individual, they both will be expressed. With respect to IA and O, IA is dominant, and likewise in IB and O, both of them, like IB is dominant. So they both share a dominant characteristic with O.
Okay, well done. So, on the basis of antigen presence or absence, that is, blood group O is also known as the universal donor. Can you justify the statement why it is known as the universal donor?
Because it does not have antigen on its RBC, it does not have either A or B. So whenever if a person who is having blood group A, if he or she donates blood to any person, that person's antibodies are not going to attack his antigen because he does not have any antigen present on the RBC. So he can be called universal donor, although the practice is not continued now. We tend to prefer that the person of the same blood group is donating the blood to the patient.
Okay, well done, well said. Sneha, there is a term that is epistatic. What is the meaning of epistatic interactions?
I'm not able to recall.
Okay, have you heard about the pleiotropic effect?
Yes, sir. What facts, sir?
Pleiotropy.
Pleuritic effect. Yes, sir. Yes, sir. Uh, I got fully confused now. No, sir, I don't recall it.
Okay, no issue. Don't
I
feel
read
bad.
all
It
of
happens
them.
with
I don't know
everyone.
what.
So don't feel down. Okay, Sneha, there are certain disease which is associated with the chromosomal abnormalities. So have you heard about the that is Down syndrome, Patau syndrome, and Edward syndrome?
Yes, sir. I've heard of Down syndrome,
So
in which
what is the genotype of Down syndrome, and what kind of abnormalities in case of a Down syndrome is there?
the 21st chromosome shows trisomy, as far as I can recall, in Down syndrome.
So what is the trisomy?
I think um, um, it will be like for each chromosome, there are like two, how do I explain it? Uh, we are diploid organisms, so we are tend to have two sets of chromosomes for each feature. One we receive from mother's gamete and the other from the father. But in this case, they will be having three alleles for the particular chromosome.
Okay, do you have idea about the nullisomy, monosomy?
Yes, sir. Uh, in Nalisomy, like XO is what example I can think of. The mother has transferred the X chromosome to the offspring, whereas the no kind of chromosome was present from the father, so it gives rise to XO.
Sneha, for these kind of
That is Nalisomy.
okay, Sneha, for these kind of abnormalities, there are mathematical expression. There is a mathematical formula like as nullisomy, monosomy, trisomy, tetrasomy. Can you just elaborate the formula for nullisomy, monosomy, trisomy, and tetrasomy?
No, sir, I do not recall it.
Okay, no issue. Uh, Okay, Sneha, in genetics, there are certain that is abnormalities regarding to the chromosomal variations and chromosomal number. So in case of that, that is, suppose there is a term known as the euploidy and aneuploidy. So what is the meaning of a ploidy?
Suppose the organism is diploid, so it is known as like it will have two N, which is known as the like the number of set of alleles for particular gene present is known as ploidy.
Set of alleles.
Okay, no, more alleles can be present
So
in
if I
diploid
ask
or
to you, that
less
is,
than.
in terms of ploidy, what we are then?
We are diploid, but yeah, I know that more alleles for particular gene can be present in human as well. I just recalled that. Uh, the chromosomes which are present in our body have there are two sets of chromosomes, one which we have received from the father, one from the mother. So that is why we are diploid. But I'm not able to get it how the plotting varies.
Okay, don't feel bad,
I've
and
made a hotspot.
just calm down. Sessions are going to be so long, so there will be some possible questions right now. Okay, have you heard about the linkage or linkage group?
Yes,
What is linkage group?
I don't, sir. I'm not able to recall anything.
Okay, 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 no issue. uh, Sneha, in Mendelian genetics, there is a that is dihybrid and monohybrid cross, right? So in dihybrid cross, we generally study
Yes,
the
sir.
example of a law of independent assortment. How the law of independent assortment opposed to the linkage process? What do you mean by the law of independent assortment?
In Mendel's case, what happened is whatever gene characters, whatever phenotypes he had chosen, they were on the separate uh, parts of the chromosome. Like either the location was different or either they belong to different chromosome. So that is why he was able to exploit it. But in case of linkage, what happens? Two or more genes are present close enough, and that is why one phenotype is controlled by like two or more genes. So
Okay,
that is how it gets
no
the
issue.
term linkage because the
Sneha, I just
phenotypic expression is linked.
okay, Sneha, I just want to know, in law of independent assortment, what is the meaning of a law? What is the meaning of independent assortment?
Because he had chosen two phenotypes, like one was wrinkled and round seeds, and the other one was yellow and green pod color, I think. So both of the characters were going to the offspring without getting intermixed. Like they were following the law of independent assortment. They got assorted individually. None of the assortment was mingling with any of the passage of both of to the offspring.
Okay, well done. Sneha, can you just elaborate the five different types of vector used in the gene cloning purpose?
Five different types of vector. Um, in case of plant, animal, anything.
Any case, just five different types of vector used for the cloning purpose.
Okay. In case of plant, I can recall one is bacteria mediated, that is Ti plasmid and Ri plasmid. And in case of plant for virus, I remember since most of the RNA plant viruses are having RNA genome, so there are two. One is Gemini virus, which has single-stranded DNA, and the other is Polymovirus, which has double-stranded DNA. Both of them are used as vectors in case of plants.
Okay, okay. Sneha, as you are a MSc, that is biotechnology candidates, right? So, in the field of biotechnology, which personality have influences more to do something in field of the biotechnology? Can you name the personality? Whatever the researcher or the scientist or the
So I'm not able to hear your question.
Yeah, so being a biotechnology student, there are so many that is persons who have a very significant role in the development of a biotechnology. So my question is that among all those famous personality, which personality have influences more you, that is to do something for the biotechnology field?
Um, for
Okay,
me, I think it
okay,
would
okay,
be
take it.
Barbara McClintock,
Yeah.
who had worked on jumping genes.
Okay, Sneha, take it easy. Just tell me the five persons who are involved in the development of biotechnology. Name of the five persons.
Um, first would be the first would be the person who came up with the structure of DNA, that is Watson and Crick. And along with that, I think a lot of credit has to be given to Rosalind Franklin, who did the initial work, and it was possible because of her. Second, I think Barbara McClintock, as already I mentioned, she was the person who was able to discover the jumping genes or the transposable elements. Um, and one more person I think would be um,
Okay, no issue, no issue. Leave it. That is, you mentioned Barbara McClintock for the jumping gene. Uh, Jumping gene is also known as the molecular parasite. Can you justify why the jumping gene or transposable elements are also known as the molecular parasite?
molecular parasite. Um, they have one enzyme transposase, and they can jump from one particular site to the other site. And maybe we are relating it with parasite because as virus, they can also integrate their particular genomic material at any place in the chromosome. So maybe that is why. I'm not sure.
Okay, Sneha, what was the contribution of a Michelson stall in the molecular or in the biochemistry or in the that is biotechnology? Can you elaborate the contribution of a Michelson stall?
The
Yeah.
Messerson stall did the experiment which told us that semi-conservative mode of replication is followed in DNA, if I'm not wrong, right? I'm not sure about it. It is stall?
Semi-conservative, right? There,
Yes, sir.
there are certain other modes of DNA replications like as conservative, semi-conservative, and other one
Dispersive.
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 So, can you just tell me the example of organisms or the conditions or the cells where other two types of mode of replications can happen?
The other two types can happen.
Yeah.
No, sir, I don't know about this.
Okay. So what is the mean of a semi-conservative mode of replication?
Semi-conservative mode of replication means that one of the parental strand is transferred to the offspring, and the other strand is newly synthesized,
Okay. So can you tell me the
the offspring.
okay, Sneha, can you tell me the name of the technique which was used to prove that DNA replication is semi-conservative in nature in the Michelson stall experiment? Yeah.
They had radio labeled nitrogen with like I think a heavy isotope of N15 was used, and later what they did is they grew the parental strand. They allowed the bacteria to grow in. Firstly, they had grown it in N15 media, and in the next generation they replaced it with 1040, and subsequently they did it for a third time and fourth time, and then they used cesium chloride gradient centrifugation to find out bands. So in the first generation, they got two bands, wherein in the first generation they got one band, whereas both of them were M15. Then for the second generation, they got a hybrid, like N15 and N14 both.
And what about the third generation?
Okay. In the third generation, they found somewhat in the middle one more, like which was a like a heteromer of N15 and N14 both, which gave a new band in between both the bands, and that
Okay,
led to the conclusion that semi-conservative.
okay, Sneha, if I ask to you, that is, what was the ratio in third generations of installation experiment with respect of nitrogen 15 and nitrogen 14, that is parental and new. What was the ratio in third generation?
In generation. Most of them were N14. So only I think if we get four, so two strands will be completely made from N14, and the other two strands, the other two they will will be one. The ratio will be 1:1.
Okay, Sneha, moving. Okay, Sneha. Apart from this, currently, what is your work? That is, what are you doing currently?
Jobs I am working as a creative writing mentor.
Okay, Sneha, tell me just simple thing. Why you think that you are a good and fit candidate for the CPTM post?
So I could not hear your question.
Okay, my question is that
Just,
why you think that you are a good
can you
and
please repeat it?
fit candidate for the CPTM post?
Yes, sir. One of the prime reasons that I think this is because I take after this post came out, I also completed a certification course by WIPO, which is World Intellectual Property Organization. Um, I take keen interest. That would be the primary reason. The second thing is that since I have a background in creative, which also involves a lot of research, and in case of job of patent examiners, also I have to go through the prior art, as in whoever has first published their patents regarding this. That would also require some research. Thirdly, I think my knowledge in biotechnology, since I come from the background of biotechnology, that would also help me. And as a biotechnologist, I could just keep in touch with all the new inventions or innovations happening in the field of biotechnology. But as an examiner, I get to see the nitty gritties of the working mechanism of the different kind of innovation and inventions that are going to take place. And apart from that, I think I'm a very hardworking person. So I'll be an asset to the organization.
Okay, good. Well said. Sneha, in RDT technology, there are certain terms like as isosizomers and neosizomers. So, can you tell me that what is the concept of the isosizomers and neosizomers?
Isosizomers are those kind of restriction enzymes wherein the um, restriction site and the restriction cutting site is same. And in case of neosschizomers, these are different enzymes, but they have the same restriction site, but they cut differently.
Okay, uh, and in recombinant DNA technology, restriction endonuclease play a very significant role. right? And as you know, that is it's a kind of a endonuclease enzyme. So my question is that why restriction enzyme is especially known as the restriction enzyme, not to the simply endonuclease? What is the mean of a restriction endonuclease? Yeah.
Endonuclease, it is called endonuclease since it is cutting uh, at or near the restriction site.
Now, what is the mean of a restriction?
It will, in the DNA strand, most of the exonuclease will cut from the ends of the strand. So they are known as exonuclease. Exo as in exterior and endo as in interior. So they will cut in near or at the restriction site. So they are known as endonuclease.
No, no, it's a fine with the endonuclease. My question is that why it is not simply known as the endonuclease? It is known as the restriction endonuclease, RE, restriction endonuclease enzyme. So, what is the mean of restriction? Yeah.
Because it will not just cut anywhere, but it will cut at a place which is known as the restriction site. There is a specific sequence which it recognizes and cuts there, which is known as the restriction site. So they termed it as restriction endonuclease.
So each and every kind of a restriction endonuclease has a specific site. That means they will only act to the that specific sequences, but they doesn't. That is, they do not use suffix of a restriction endonuclease. So why it is especially for the restriction endonuclease? Okay, Sneha, what is the source of a restriction endonuclease
I'm
enzyme?
not aware about that, sir.
Okay, leave it. What is the source of these restriction?
It is called from bacteria. Bacteria uses,
Yeah.
I think my voice is reaching later, right? Uh, the bacteria had evolved a system in order to escape the attack by virus. So they had developed restriction enzymes,
Okay.
which cuts the specific sites at the nucleic acid of the virus.
Okay, suppose that is today we know that resistant enzyme has a very unique property. That means it will act on any kind of a source of DNA, that is from RNA, plant, bacteria, animal, etc. So if the bacteria uses for the digestion of a viral DNA, then how it prevent itself DNA from the digestion of a resistant enzyme?
Because the bacterial DNA, where the restriction site is present, is methylated. Because of the methylation, the restriction enzymes cannot act on its own DNA.
Very good. My next question is that is how methylation means that is addition of a CH3 group, that is methyl group. So how a single molecules can act as a prevention of acting of that restriction enzyme? What is the concept behind it? How a single molecule of methyl will disrupt all the mechanism of a restriction enzyme?
Um, as per my limited knowledge, I think the CH3 group, which is attached to the restriction site, they might provide some steric hindrance for the endonuclease to act at that particular place. And it does not allow it to reach or sit there. So that is why it is not able to cut it.
Okay, very good. So the restriction enzyme is also a kind of enzymes, and as we know, each and every kind have their specific active sites, and the enzymes can only bind to that substrate which have the complementary shape of that active sites. So, Sneha, in the in the enzyme and substrate binding, there are certain chemical bond involved. So, can you tell me the chemical bonds involved in the substrate and enzyme binding?
All of them will be weak bonds like non-covalent bonds, hydrogen bonds,
Okay, non-covalent bonds like it's a hydrogen bonds and
and non-covalent bonds, van der Waals forces of attraction. and hydrogen bonds.
Okay, Sneha, suppose if there is a covalent bond interactions between the substrate and enzyme, then what will happen?
Then it will not. The enzyme is supposed to do its job, get attached to the enzyme, make specific changes in its active site, and change it to a product. But if there is covalent interaction between the enzyme and the substrate, then it is going to be a permanent kind of bond. So the enzyme might not be able to convert it into product.